unfold, cracking the code. Genetic secrets will be told, cracking the code.
The script that's written in our genes has an alphabet of only 4 letters. It is the irregular and utterly unique sequence of these letters, 3 billion of them, that makes us human beings, and each of us, unless we have an identical twin, different from each other. We have now learned how to read, exploit, and even rewrite the genetic code within ourselves and all other forms of life. These explosive developments in genetics and biotechnology are bringing about a revolution in science, medicine, and agriculture. As it expands in pace and power, this revolution promises to bring even greater changes in the way we live and how we create new life. It will also present us with difficult new choices and dilemmas, and it is the subject of this series. How did we arrive at this point? Cracking the code of life is the culmination of a quest that is as old as human observation. We begin with the ancient puzzle of heredity. From the beginning, it was clear that in some cases, physical traits are passed down from parent to child, sometimes on through the generations. But in other cases, a child might bear little resemblance to either parent or to its siblings, or more of a resemblance to a grandparent than to a parent. There seemed to be no consistent pattern within the visible complexity of life. The ancients believed in divining or reasoning their way to the facts, rather than using experiment and observation. Most of these guesses about biological inheritance were wildly wrong, yet some of them survived into the modern era. The ancient Greek philosophers, like Aristotle, thought that the male parent was more important than the female in determining heredity. Perhaps this was because they viewed the semen of the male as resembling the seed of a plant, and a seed usually develops into a plant like the one it came from. They saw the reproductive role of the female as akin to the soil which nurtures the plant. But that theory had an obvious flaw. Half of all children turn out to be girls. A good thing, but hardly a chip off the old male block. To get around that one, Greek philosophers had to come up with a special theory about the origin of gender. Some thought the sex of the child was determined by which testicle the sperm came from. The one on the right, producing boys. The one on the left, producing girls. That still didn't allow for any inheritance from the mother.
Give me a break.
This belief was advanced by the influential Roman physician Galen, and it persisted within medical circles until the 19th century discovery of the mammalian egg. Observation, as opposed to speculation, started with the breeding of animals, which began long before the Greeks. Over the course of time, farmers and shepherds noticed which traits were passed from either of the parents to their offspring, and gradually learned how to select out and crossbreed those animals with the most desirable traits. The crossbreeding of plants came much later. In fact, it wasn't until the 1690s that it was discovered that plants have sex organs and the ability to cross-fertilize. In the 1700s, the Swedish scientist and physician Carl Linnaeus undertook the monumental task of cataloging and classifying every known form of life. Linnaeus was intrigued by plant sexuality and categorized plants on the basis of their sex organs. To others, these were a source of amazement and even scandal. To some moral critics, so much pollen and so few seed chambers suggested female promiscuity. Linnaeus's lasting achievement was a two-part system for naming each species, which is still in use today. A major controversy among scientists of the time was whether species are unchangeable or whether they can evolve. Many botanists firmly believed in the biblical teaching that species were fixed at creation. Linnaeus disagreed. He was sure that crossbreeding two different plant species would produce a new intermediate one, but he didn't put his theory to the test. A contemporary of his, the German botanist Joseph Kuhlreuter, decided to settle the issue with the first true experiments in genetics. Unfortunately, much of his work was sabotaged by his disgruntled gardener, Saul. Kuhlreuter began by crossbreeding two different species of tobacco plant. These plants are called the parent or P generation. He then planted the resulting hybrid seeds, which is called the F1 generation. These seeds produced a new type of plant that retained some of the features of both parents while losing others. But the real test of whether this hybrid was a new species was if it could reproduce itself. In fact, cross-fertilizing the F1 generation produced only a few new seeds. Kuhlreuter was not surprised, since like many others, he believed in an unchanging nature. But he was surprised at what happened next. When he planted these seeds to produce the next, or F2 generation, some of the missing traits from the P generation reappeared. This turned out to be an important clue in solving the riddle of heredity. But Kuhlreuter didn't recognize its significance. Perhaps he was too distracted by his gardener. Charles Darwin, the great English biologist, lived and worked in this house, south of London, for most of his life. He too was interested in the evolution of new species, which by the mid-19th century was becoming an accepted fact. Darwin noticed that domestic plants and animals gradually evolved due to breeder selection of particular varieties. He was also much taken with the idea that overpopulation would lead to competition and to the survival of the fittest. To explain evolution in nature, Darwin developed his theory of natural selection, that nature selects out for survival those variations that are best equipped for reproductive success. His book on evolution was published in 1859. It caused a sensation and made him a world celebrity. Darwin's theory of natural selection, leading to evolution, has stood the test of time. But he also wanted to explain variation, the raw material upon which natural selection works, and heredity, the way a selected variation is passed down to future generations. In that, he was far less successful. Darwin, like many other biologists of his day, mistakenly believed that reproduction is a blending process, which evens out the differences between the two parents. If so, this would tend, over time, to reduce variation. Since natural selection requires a choice between variations, blending would eventually bring evolution to a halt. To overcome this contradiction, Darwin speculated that the material of heredity, still totally unknown, was somehow altered after fertilization to restore its variability. This was a form of another mistaken belief called acquired inheritance. It holds that any changes a plant or animal undergoes in response to its environment are passed on to its offspring. It's an appealing idea, since living things clearly can change in response to a changing environment. But it confuses appearance with genetic endowment. This confusion contributed to the late start of genetics relative to the other sciences. It is not surprising that Darwin got heredity wrong. So did everyone else who tried to find an underlying pattern in a surface picture that seems so random and disorderly. But unbeknownst to Darwin or anyone else, someone was busy solving this great puzzle. In the process, he laid the groundwork for the new science of genetics. Gregor Mendel, the man who solved the puzzle of heredity, was as obscure as Darwin was famous. He was born in 1822 into a peasant family in a region of Austria that is now within the Czech Republic. He was christened Johann. His intelligence was recognized early on, and he was sent away to school and then to university. A rare event for someone from such humble origins. However, when Mendel was 17, his father was injured and lost his livelihood, which threatened Mendel's studies. One of his teachers, an Augustinian monk, suggested that he enter his monastery in the town of Brno as a way to continue his education free of financial worry. Once admitted into the order, Mendel was given the new Christian name of Gregor. Pastoral duties did not agree with him, and he took on the job of substitute teacher at the local high school. Mendel had a natural affinity for both science and for teaching it. This led him to take the permanent teacher's exam, which he flunked. It was suggested that he bone up at the University of Vienna and try again. In Vienna, he took an interest in physics, which introduced him to the science of statistics. Applying statistics to biology proved critical to his later success at sorting out heredity. After returning to the monastery, Mendel decided to combine his lifelong interest in plant breeding with his new mathematical skills. Like Darwin, he was curious about evolution. And like Kuhlreuter, he wondered if a new species could arise from the crossbreeding of two existing species. But Mendel decided to first study the variations that arise within a species. It's not as grand a topic as evolution, but it gave him the access he needed to enter the hidden world of heredity. Mendel's predecessors had focused on entire plants. Mendel wisely chose to isolate a single feature at a time and follow it down the generations. He also resolved to count his results, something that had never been done before, and to look for patterns in the numbers he came up with. Mendel searched for the ideal experimental model and found it in the common garden pea plant. It has a number of traits that come in two contrasting forms, which make them easy to spot and categorize. For instance, the pea pods are either full and smooth or shrunken and wrinkled. Stems are either short or tall. Its flower has a shape that protects against contamination by stray pollen. And lastly, it self-fertilizes. Its pollen fertilizes eggs within the same flower. This allows it to produce seeds which breed true, meaning each offspring plant is exactly like its single bisexual parent. Over the following 8 years, Mendel planted an estimated 28,000 carefully controlled crosses between different varieties of pea plants. He explored every possible combination, sometimes for up to 7 generations, and meticulously counted the results. One of the contrasting features that Mendel studied was the color of the plant's flower. One of his true breeding varieties always grew purple flowers, another always grew white ones. Their seeds formed the parent or pea generation. As soon as buds appeared on the vines, Mendel carefully opened them and removed the stamens to prevent self-fertilization. He later took pollen from purple flowers and sprinkled it on the pistils in white flowers. He also did the reverse. To prevent contamination by wild pollen, he then wrapped the flowers with protective bags. As a further control, he moved some of the plants into the insulated environment of a greenhouse. Weeks later, both groups of cross-fertilized plants developed seeds. Mendel stored the hybrid seeds over the winter and planted them the next spring. When the plants in this first hybrid or F1 generation grew flowers, they were all purple. Mendel called this trait the dominant one. He termed the trait that disappeared in the F1 generation recessive. Mendel let these F1 hybrid plants self-fertilize and produce new seeds. The next year, Mendel planted the F1 seeds, producing the next or F2 generation of plants. This time, there were some white flowers mixed in among the purples. He was well aware that traits sometimes skipped generations, so this was no surprise. But Mendel discovered something that no one else had ever noticed before. He counted the number of white and purple flowers. In similar fashion, he counted the results of crossing 6 other contrasting traits through to the F2 generation. When he worked out the ratio of dominant to recessive, in each case it came very close to 3:1. Mendel had uncovered a pattern of inheritance, something that had eluded everyone from Aristotle to Darwin. This was a great discovery in itself, but Mendel also wanted to explain it. Using just his data and his imagination, Mendel came up with a model of how heredity works that remains the cornerstone of genetics. He concluded that life is not transmitted in one piece, as most people assumed, but in discrete particles, which he called factors, and which we now call genes. Using his particle model, Mendel was able to explain the great paradox of heredity, how both inheritance and variation can coexist, how like can beget both like and unlike. Mendel concluded that these invisible factors are passed along unchanged, which accounts for inheritance, but that sexual reproduction leads to new and different combinations of them, which accounts for variation. Mendel theorized that a trait is determined by not one, but 2 factors or genes. These factors also come in dominant or recessive forms. But Mendel made an important distinction between the nature of the trait, which we call its phenotype, and the nature of the underlying pair of genes, which we call its genotype. For his experiment with purple and white flowered plants, there were 2 possible phenotypes, dominant purple or recessive white, and 3 possible genotypes, 2 dominant genes, in which case the phenotype is purple, 2 recessive genes, in which case the phenotype is white, and 1 of each, in which case the single dominant gene causes the phenotype to be purple. Mendel imagined that in the production of a sperm or an egg cell, the gene pair is split apart, with each gene ending up in a separate germ cell. Mendel called this process segregation. He also imagined that the direction of segregation is totally random. It can just as easily happen the other way around. So for any particular germ cell, which one of the gene pair it receives is totally up to chance, like the flip of a coin. This became Mendel's first law of genetics, the law of random segregation. This is how Mendel explained his results. His parent plants, the P generation, bred true for color. This meant they each had a pair of matching genes. The purple plant had 2 dominant genes, and the white one had 2 recessive genes. A genotype with two copies of the same gene is called homozygous. When the parent plants were cross-fertilized, they each contributed one flower-colored gene to the next generation. Since the purple parent had only dominant genes to contribute, and the white parent only recessives, each offspring had a genotype with one of each. This kind of mixed genotype is called heterozygous. A dominant gene trumps a recessive gene, so every plant in the F1 generation had purple flowers. The purple F1 plants were then allowed to self-fertilize to produce the next, or F2 generation. According to the law of random segregation, for any particular germ cell, the odds are even as to which gene it receives. So about half the sperm and egg cells carried the purple gene, and about half the white one. As a result, for any given fertilization event, there are 4 equally likely outcomes in terms of genotype. A purple pollen can fertilize a purple egg. A purple pollen can fertilize a white egg. A white pollen can fertilize a purple egg, and a white pollen can fertilize a white egg. The homozygous purple genotype produces a purple phenotype. The homozygous white genotype produces a white phenotype. Because the purple gene is dominant, both heterozygous genotypes produce purple phenotypes. What we are left with are 3 purple plants and 1 white one, the same 3-to-1 ratio that Mendel found in his various F2 generations. His experimental results were now explained. The 6 other contrasting traits that Mendel tested also conformed to the law of random segregation. But Mendel was curious about what would happen when 2 of these traits occurred together in the same plant. For instance, one of the traits he studied was the color of the seed. Yellow was dominant and green was recessive. Another trait was the shape of the seed, round being dominant and wrinkled being recessive. Mendel cross-fertilized homozygous plants whose seeds were always both yellow and round with plants whose seeds were always both green and wrinkled. Would color and shape each follow its own pattern of random segregation? Or would the 2 traits somehow melt together? Think of a slot machine with 2 spinning wheels, one representing the seed's color and the other its shape. Each spin is like a segregation event. Mendel wanted to know if the 2 wheels spin independently of each other, or if they are somehow linked. Mendel crossed hundreds of plants with double traits. Again, he carefully counted the results and compared them to the numbers he would expect if the traits continued to segregate out in random fashion. The 2 sets of numbers matched, proving that each trait did sort itself independently of the others.
Well, congratulations.
Mendel now had his second law of genetics, the Law of Independent Assortment. Sadly, Mendel never got a chance to celebrate or be celebrated. His momentous discoveries were totally ignored in his lifetime, even though about 120 copies of the journal containing his paper were sent to libraries around the world. In contrast, Darwin, his contemporary and fellow giant of biology, won great fame within his. Perhaps if Darwin had heard about Mendel and his laws, much would have changed for both of them. It almost happened. Mendel mailed a copy of his paper to Darwin, but apparently Darwin never opened the letter. Mendel died in 1884 at age 62. It took another 16 years for the world to finally learn about him.
Oh, why, dear God, did you make it so complex to understand the offspring that result when there is sex? But there is one monk among us who can tell how it all works. We feel like jerks next to Gregor Mendel.
The answer's in my garden where I've planted different peas and sprinkled pollen as I pleased and counted out the progeny.
What did you discover in your garden with your peas about those factors we can't see but which explain our family trees?
Here's the news, they come in twos, they separate, it's up to fate. If an egg or a sperm has a trait that will dominate.
Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate.
And when they join together, my forecasting's most impressive. Bet you 3 times out of 4 I'm right unless they're both recessive.
To what do you owe your success?
To counting and my green thumb. But where these hidden factors are, well, that I cannot fathom.
Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate. Later on the world was odd at what he learned from those pods. But back then no one hurrahed Gregor Mendel but his God. Back then no one hurrahed Gregor Mendel but his God.
From life's page, how did we get to this new stage? To solving what was once life's mystery. Would you like to know from whence you've sprung? Would you like to stay forever young? Would you like to shape your own heredity? We're learning how to pull upon the strings to rewrite the script from which life itself springs. Cracking the code. Genetic futures will be foretold. Cracking the code. Genetic power about to explode. Cracking the code. Genetic mysteries to unfold. Cracking the code. Genetic secrets will be told. Cracking the code.